Hello students, we have already learned about the features of a cell that are plasma membrane, nucleus and cytoplasm. Let's take the example of an ecosystem, where some factors, both biotic and abiotic exist, without which an ecosystem can't function properly. Similarly, if we call cytoplasm an ecosystem, then its components are organelles without which a cell will not function properly. Now, let's study the most important constituents of the cell. That is, organelles. Every cell has a membrane that separates its contents from the outside environment. Large and complex cells, especially those seen in multicellular animals, require a high level of chemical activity to support their complicated structure and function. These cells use membrane-bound tiny structures, or organelles, within themselves to maintain these various processes apart from one another. This is one of the characteristics that distinguishes eukaryotic cells from prokaryotic cells. Some of the organelles we are going to study are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, plastids, vacuoles. Let's understand them one by one. The vessel-type structure inside the cell is endoplasmic reticulum. It consists of a large network of membranous sheets and tubes. It looks like long tubules or round or oblong bags or vesicles. Endoplasmic reticulum is basically the manufacturing unit of the cell. If you remember this, it will be easy for you to remember what is the role of endoplasmic reticulum as an organelle in the cell. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, RER, and smooth endoplasmic reticulum, SER. Rough endoplasmic reticulum appears rough under a microscope because it has ribosomes adhering to its surface. Ribosomes, which are found in all active cells, are the sites of protein synthesis. Using the endoplasmic reticulum, the proteins are subsequently sent to various locations within the cell based on their needs. The smooth endoplasmic reticulum helps in the manufacture of fat molecules, or lipids, important for cell function. Some of these proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. The functions as a channel for the movement of materials, particularly proteins, between different regions of the cytoplasm or between the cytoplasm and the nucleus. The also serves as a cytoplasmic framework, providing a surface for some of the cell's biochemical activities. SER plays an important part in the detoxification of various toxins and drugs in the liver cells of the vertebrate group of animals. Let's now explore the Golgi apparatus and its functions. The Golgi apparatus was first described by Camillo Golgi. It consists of a system of flattened sacs that are arranged approximately parallel to each other in stacks called cisterns. Cisterns are bound by membrane. There are various vesicles associated with Golgi apparatus, such as transport vesicles and secretory vesicles. The material synthesized in the is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. Hence, it is the packer and transporter of the cell. It basically stores, packs and modifies the products in vesicles. Then, it transports the products inside and outside the cell using vesicles. The Golgi apparatus is also involved in the formation of lysosomes, as some vesicles become lysosomes. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus. Now, let's study the suicidal bags of cells. Lysosomes. One must wonder why we call lysosomes by this name. So let's explore its answer. Lysosomes are a kind of waste disposal system of the cell. These aid in the cleaning of the cell by digesting foreign material as well as worn-out cell organelles by breaking complex substances into simpler substances. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. Lysosomes may burst and enzymes digest their own cell during a disturbance in cellular metabolism, such as when the cell is damaged. As a result, lysosomes are often known as a cell's suicide banks. Let's discuss the powerhouse of the cell, that is, mitochondria. Mitochondria is known as the powerhouse of the cell. Mitochondria have two membrane coverings. The outer membrane is porous while the inner membrane is deeply folded. These folds increase surface area for ATP-generating chemical reactions. The energy required for numerous chemical reactions required for life is released by mitochondria in the form of ATP, adenosine triphosphate, molecules. Mitochondria are unique organelles in that they contain their own DNA and ribosomes. As a result, mitochondria can produce some of their own proteins. Have you ever thought about the green color of the leaves? It is the organelles plastids that give pigment to the plant cells, hence leaves are green in color. Now, let's discuss it in detail. Plastids are the kitchen of the cell. Plastids are only present in plant cells. They are called the kitchen of the plant cell because they are able to make their own food by using the sunlight. So, as in the kitchen food is prepared, similarly in plant cells, food is prepared due to plastids. This special type of cell organelle helps the plant in photosynthesis, storage and also provides color. We have two types of plastids: chromoplasts, colored, and leucoplasts, white or colorless. Chromoplasts impact specific colors to the flowers, fruits, and leaves. Leucoplasts are primarily organelles that store materials like starch, oils, and protein granules. Chromoplasts containing the pigment chlorophyll are known as chloroplasts. The chloroplast's internal organization consists of multiple membrane layers embedded in a substance known as the stroma. In addition to chlorophyll, chloroplasts contain a variety of yellow or orange pigments. Plastids have their own DNA and ribosomes just like mitochondria. Vacuoles take up the most space in cells. Why is it so? Let's discuss it in detail. Vacuoles are fluid-filled organelles surrounded by a membrane. Animal cells have small-sized vacuoles, whereas plant cells have large vacuoles. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. It provides turgidity and rigidity to the plant cell. You can remember the function of the vacuole by calling it the storeroom of the cell, as the cell stores many things inside the vacuole. It acts as storage sacs of the cell and stores food, water, sugar, minerals, and waste products of the cell. Many important substances for plant cells' life are stored inside the vacuole like amino acids, sugars, various organic acids, and some of the proteins. In single-celled organisms, like the amoeba, food vacuole contains all that amoeba consumes. In some other single-celled organisms, vacuole play the role of expelling water and wastes also. Till now we have discussed so much about the cell and its organelles. Let's now discuss how these cells form. So, let's study cell division. In order for organisms to develop, replace old, dead, or injured cells, and generate gametes, such as eggs and sperms which are essential for reproduction, new cells are formed. Cell division is the process by which new cells are formed. Cell division is classified into two types: mitosis and meiosis. Mitosis refers to the process of cell division in which the majority of cells divide for growth. In mitosis, each mother cell divides to form two identical daughter cells, having the same number of chromosomes as of mother cell, and these cells help in growth and repair of tissues in organisms. Meiosis involves two consecutive divisions, which result in 4 new cells instead of 2. But here these new cells have half the number of chromosomes than that of the mother cell. Gametes divide by the process of meiosis. So that concludes this video, as we have covered all of the topics in this chapter. Now it's time for you to go and start practicing so that you can answer every question in the exam and get a perfect score. This is only possible if you keep revising by reading notes from EduRef, taking tests on the same topics from EduRef, and going over the chapter again and again. Thank you for watching the video.